Hello, everyone. Thank you for being here for this July 27th ISEE North American Chapter webinar. We're really excited. This is our first SNRN webinar that we are sponsoring. We are joined by Dr. Max AUNG, who is an associate research scientist at the UCSF Program on Reproductive Health and the Environment, or PRE. Prior to joining PRE, Max completed doctoral training in environmental epi and postdoctoral training in biostatistics and precision health science, data health science at the University of Michigan. Max is an alumnus of the RWJ Health Policy Research Scholars Fellowship, as well as the Agents of Change in Environmental Justice Fellowship. Two really exciting fellowships folks should check out. In his current role at PRE, Max is motivated to build a research program that focuses on environmental justice and integration of cutting-edge data science and statistical tools to investigate the biologic mechanisms linking the prenatal exposome to adverse reproductive and developmental health outcomes. So Max is really passionate about academic service. He currently serves on the editorial board for the Journal of Exposure Science and Environmental Epidemiology and the Student New Researchers Steering Committee for ISEE, which is what we are sponsoring this webinar on today. So we're very excited, Max, to have you here with us. Please go ahead and share your screen. Max will give us a 30 to 40-minute presentation, and then we will have some time for discussion. So let's hold questions until the end here. Okay, thank you all.
Awesome. Thank you so much for that really great introduction, and good morning, everyone. It's such a great pleasure for me to begin this new tradition in the ISEE North American chapter of hosting this annual Student or New Researcher Network webinar. And I'm excited to use this space today to talk about some of the research that I've been focusing on more recently. So I wanted to start off by just taking a moment to let the audience know that just as Joan said, I'm serving on the ISEE SNRN Steering Committee, and I'm joined by an incredible group of scholars across the world. And for the past 2 years, we've been developing programming and strategies to support students and new researchers within the society. And we will be hosting multiple exciting panels at the upcoming ISEE conference. So please reach out if you want to get more involved or learn more about some of the things that we're working on. So for my presentation today, I will be covering 3 main themes that have emerged from my work this past few years. I will be talking about some of the initial work that we've conducted evaluating bioactive lipid biomarkers and their utility for prediction of particularly pregnancy outcomes. Another theme of my talk is going to dive into the work we've been doing to explore these bioactive lipids as potential mechanistic signals of the prenatal exposome. And finally, the third theme of my talk is going to focus on some more recent work that we've been doing by expanding our knowledge of bioactive lipids to increasingly high-dimensional lipidomic scales. So when I began and pursued my doctoral and postdoctoral training at the University of Michigan, I had the privilege of building some really awesome collaborations with mentors and colleagues and conducting research in 2 well-characterized prospective birth cohorts. The first is the Life Codes cohort, which is a nested case control study of preterm birth based in Boston, Massachusetts. And these participants were recruited between 2006 and 2008. And then the other cohort is the Puerto Rico Protect cohort, which is a super fun research program cohort that's focuses on evaluating environmental risk factors in historically neglected communities in Puerto Rico. And participants in this cohort began recruitment in 2010. So through these cohorts, my research has predominantly focused on understanding risk factors of maternal and children's health. And in these cohorts, the phenotypes that I've been focusing on exploring risk factors for include preterm birth, as well as some collaborations with Kelly Ferguson at NIEHS, focusing on fetal growth, and more recently collaborating with Emily Zimmerman at Northeastern University to study child neurodevelopment in the Protect cohort through leveraging some new technology to assess infant non-nutritive suck. So across both of these cohorts, we've also had the capacity to explore extensive biomonitoring of chemical biomarkers and trace metals. And we've also had the capacity to explore emerging assays to quantify endogenous biomarkers of intermediate effects. So this data has really allowed me to develop some exciting new skills in mixtures methodology, as well as developing creative frameworks for conducting data integration across complex data frames. So among the chemical biomarkers in the live coats cohort, we've had the capacity to measure 4 different classes. And this includes phthalates, phenols and parabens, trace metals, and polycyclic aromatic hydrocarbons, all of which were measured in urine samples. And you can see in the corresponding images here some of the common sources of human exposure to these chemicals and trace metals. And based on the mechanistic literature, we know that these chemicals can act through multiple different biological pathways upon human exposure. So some examples include interaction with circulating immune system, which can lead to the upregulation of inflammatory molecules. These toxicants can also interfere with receptors in enzymatic cascades, which can interfere with metabolism of macromolecules. And in addition to this, these toxicants can interfere with the intracellular balance of reactive oxygen species, which can result in local and systemic oxidative stress. So these are some broad examples of biological pathways, and I've outlined some of these chemicals that we've measured, but now I'll highlight some of the particular endogenous biomarkers that we've been focusing on most recently to sort of try to capture these particular biological pathways. So this figure here I cited from, it was created from one of my colleagues at NIEHS, Barrett Welch, who nicely visualized some of these lipid biomarkers that we've both been investigating these past few years. And we started with an exploratory cross-sectional study from the live coats cohort, and we quantified these biomarkers in plasma samples using liquid chromatography coupled to mass spec. And you can see here in the center of the figure that there are these parent unsaturated fatty acids, such as arachidonic acid and linoleic acid. And in the periphery, there are these colored boxes, which represent the secondary eicosanoid metabolites that are metabolized from these parent compounds. And so the concentration of these eicosanoids are governed by conserved families of enzymes, which includes cytochrome P450s, lipoxygenases, and cyclooxygenases. And what's really, interesting about these biomarkers is that they have diverse physiological functions that range from cardiovascular functions such as vasodilation and vasoconstriction, as well as angiogenesis. In addition to this, these bioactive lipids can stimulate inflammation as well as resolve inflammation. And some of these bioactive lipids are also found within exosomes, which are used for cell-to-cell communication, and particularly during pregnancy, they are important for fetal and maternal crosstalk. So what I've found to be really nice about these biomarkers is that we can leverage this biological hierarchy, particularly by these enzymatic groups, and take advantage of that correlation structure for statistical modeling and downstream analyses. So one of the first studies that I conducted with these biomarkers was to evaluate their associations with spontaneous preterm birth. And in this particular setting, we defined this as either spontaneous preterm labor or spontaneous rupture of the membranes. So this forest plot here illustrates the associations that we observed with each of these bioactive lipids and the parent unsaturated fatty acids. So we see from this figure here that several of the bioactive lipids and the parent compounds across the board are associated with higher odds of spontaneous preterm birth. And I'll also note that after adjusting for multiple testing and false discovery using the Benjamini-Hochberg method, nearly all of these associations were robust to a false discovery threshold of 0.1, which means that we have even greater confidence that these associations are true associations and robust to false discovery. Another interesting part of this study was that we split our data set into training and testing data sets to evaluate the predictive capacity of these biomarkers. And we leveraged both linear approaches such as elastic net, which I'll explain in more detail later, and also machine learning approaches such as random forests. And we determined that among all of the different bioactive lipids, the lipoxygenase metabolites and the cytochrome P450 metabolites yielded the greatest prediction accuracy in classifying spontaneous preterm birth compared to the other biomarkers. So overall, these findings were very exciting because they have major clinical implications for early detection of preterm birth, and perhaps they may be important for other maternal and fetal outcomes during and after pregnancy, which we are looking forward to exploring in future follow-up studies. So building from that previous work, I then began to explore a follow-up study and integrated that extensive exposure panel of chemical biomarkers to explore both single pollutant and multi-pollutant associations with these bioactive lipids and other biomarkers of inflammation and oxidative stress and angiogenesis. And the reason why these associations need to be estimated is that once we do this, we can begin to develop a more comprehensive picture of these intermediate mechanisms of chemical toxicity, and then we can use those mechanisms to link to different health outcomes such as preterm birth and child development. So this figure here illustrates the results from the single pollutant models, and I'll orient you to the interpretation of this figure. So this is a heat map where each cell represents a single pollutant association, such that the black cells are positive associations and the blue cells are negative associations. The additional layer of information in this figure is that the darker shaded cells indicate smaller p-values and more significant associations. You'll also see that there are some white X's in some of the cells, and that indicates associations that are robust to false discovery. So at a very high level, one of the major takeaways that we see from this figure is that there appears to be unique signatures of associations between the different chemical classes and the different endogenous biomarkers. Now, when we zoom in, particularly for the phthalates and polycyclic aromatic hydrocarbons, we see that they exhibited the greatest positive signals with the cytochrome P450 derived bioactive lipids. And when we reviewed the mechanistic literature, we found quite a bit of biological support of these findings. For example, in vitro studies have reported evidence that exposure to parent phthalates and their metabolites can inhibit the functions of cytochrome P450 enzymes, as well as the PPAR gamma enzyme. And inhibition of these enzymes may potentially result in increased substrate and metabolite concentrations, reflecting some of these associations that we observed. For the polycyclic aromatic hydrocarbons, these chemicals can potentially bind to the aryl hydrocarbon receptors, and they can also interact with transcription factors such as NF-kappa B that are important for regulating the expression of inflammatory markers such as cytokines. Additionally, mechanistic studies have also demonstrated that PAHs can form DNA adducts, which can also influence gene expression of several enzymes, including cytochrome P450. So also zooming into the phenols and parabens, we observed largely negative signals in each of the different pathways, the enzymatic pathways. And, particularly with the metals, we saw either mostly negative or sometimes an even 50-50 split of associations. And we learned from in vitro models that chlorinated phenols particularly can alter cytosolic calcium concentration levels which can influence calcium-dependent metabolism of fatty acids. And the metals have also been shown in animal models to inhibit calcium-dependent enzymes as well. And particularly essential metals such as manganese have been shown to be potent ligands or cofactors of enzymes such as lipoxygenases. So as such, the intracellular concentrations of metals in particular, and also these chlorinated phenols, can interfere with some of these enzymatic pathways. And whether or not these mechanistic studies indicate either inhibition or activation of a given enzyme, I will underscore that it is still really difficult to determine a priori how exactly those mechanistic studies would translate to associations observed in epidemiology studies. But the fact that there is evidence of different molecular targets of these different chemical classes underscores that there is potential for nuances in these both directions and magnitudes of associations across the different chemical classes. Okay, so now that I've showed you some of these single pollutant associations, another exciting part of this extensive analysis that we applied is this ensemble of state-of-the-art mixtures analysis methods. And I'll highlight two that we applied in this particular study, which includes adaptive elastic net and a hierarchical Bayesian kernel machine regression. So adaptive elastic net is a regularized regression method that utilizes tuning parameters to penalize less predictive exposures. And so what that means in a complex mixture of chemicals is that elastic net evaluates which of the most important associations with which of the exposures have the most important associations with the given outcome variable. And the major advantage of this approach is that adaptive elastic net it performs very well in the presence of multicollinearity, and in particular with highly correlated exposures, this can be a very pervasive problem, especially when you're looking at multiple chemical classes. So this figure here is that same sort of heat map grid that I showed you earlier, but instead the interpretation is a little bit simpler. So here in this heat map, the black cells are simply positive signals that were selected by adaptive elastic net. White cells are no signals and complete shrinkage to zero. And blue cells are negative signals. And so just to remind you, each of these biomarkers on the y-axis here is now adjusted for all of the exposures combined in the same particular model. So the pattern that we start to see here from these findings is that the phthalates continue to be consistently positively associated with several of the eicosanoids and parent fatty acid compounds. And we also see again that signal with the polycyclic aromatic hydrocarbons that parallels the phthalates for these cytochrome P450 metabolites. And for the phenols, we see again this majority of negative signals that are across the board with the different eicosanoid enzymatic groups. And the metals exhibit either negative signals, particularly with the lipoxygenase pathway, or sort of this 50-50 split with the cytochrome P450 and cyclooxygenase pathways. So ultimately, the significance of these multi-pollutant findings is that it really helps us to prioritize biomarker signals using this whole chemical profile approach, and it helps us obtain greater confidence in those single pollutant associations that we applied that I showed you previously. And balancing the two really helps gain greater insight into some of these most important biomarker signals. So the second method that we applied was the Bayesian kernel machine regression. And this method combines multiple pollutants together into this flexible kernel function in association with a given outcome variable. So the advantage of this method is that it can model nonlinear associations, and it is particularly flexible. But one of the drawbacks is that it can be computationally intense, and sometimes when your exposure matrix is really high dimensional and you have very sparse signals, sometimes it's difficult for the kernel function to detect some of the nonlinear relationships. So the output that I was particularly interested in with BKMR is that it takes each of the exposure variables and it provides you with posterior inclusion probabilities, which is an indicator of variable importance. And from this output, we typically set a threshold of 0.5 as important. And I'll also note that in our application of BKMR, so we applied the hierarchical clustering option. So instead of getting just single chemical metabolite posterior inclusion probabilities, we also got whole class level posterior inclusion probabilities to give us some indication of the entire chemical class in relation to the particular outcome. So this figure here is another heat map that illustrates this whole class level variable importance in association with these individual endogenous biomarkers. The gray to black cells indicate positive signals where the darker cells are higher importance, and the blue cells indicate negative signals where the darker cells again are higher degrees of importance. And so these results further reinforce the observations that we reported using the single pollutant regression and elastic net models, particularly that the phthalates exhibit overwhelmingly positive and important signals with multiple bioactive lipids and their subgroup pathways, and the phenols exhibited much more negative signals across the board. And I will note that, when comparing single outcomes, not everything is totally overlapping across each of these three statistical approaches. And we documented that in the manuscript. And it was one of the discussion points that we focus on, which is that when you're applying mixtures analyses methods, it's really important to integrate an ensemble of different methods to essentially gain greater confidence in some of the associations that you observe. And particularly when you do observe some differences in findings across the different methods, it can be a function of statistical theory for the different methods and also different degrees of tolerance that the methods have for attributes of the exposure matrices, particularly sparsity of signals and multicollinearity in the exposure matrix. So recently, in addition to having measured these bioactive lipids in the life codes cohort, we've also had the opportunity to measure the same panel of molecules in a pilot sample in the PROTECT cohort as well. So this additional data across these two cohorts led us to explore this follow-up research question of whether these bioactive lipids can be used to predict spontaneous preterm birth across the birth cohorts, especially noting that these two cohorts are very different in terms of demographics and this heterogeneity in these factors can potentially be something to consider when doing these prediction algorithms. So these two figures highlight the two approaches that we applied in the prediction algorithm. So on the left, we have we applied adaptive elastic net, which assumes linearity in the predictor matrix. And we learned from applying this method that the parent polyunsaturated fatty acids yielded the greatest prediction accuracy for classifying spontaneous preterm birth. On the right panel, we used random forest, which is the machine learning tool that allows for nonlinearity and interactions in the predictor space. And we learned from that method that using all of the bioactive lipids together, rather than splitting them up into groups, yielded the greatest prediction accuracy. So these findings are interesting because they are allowing us to build the foundation for exploring different frameworks for cross-cohort predictions. And I think this will be really important in modern epidemiology studies, particularly those with multiple sites, such as the ECHO program that has multiple birth cohorts across the US with heterogeneous demographic characteristics. So going forward, we are going to be building on these approaches to explore different ways to evaluate study site differences in these downstream prediction models. So now circling back to the PROTECT cohort, another new area that we've had the opportunity to explore is a pilot sample where we conducted lipidome-wide quantification of multiple lipid classes. And this included a total of 587 metabolites. And this goes beyond those free fatty acids and includes multiple of these classes, which I've highlighted a few examples here. For example, we've learned from the literature that these have diverse biological functions. The phospholipids, for example, plays a critical role in neuronal signaling and key processes such as cellular migration. Another example includes the acylglycerides, which polymerization and catabolism is important for energy production and is a direct function of endocrine signaling. So altogether, this expansive repertoire of lipid metabolites further enhances the opportunity to characterize additional antecedent mechanisms of maternal and children's health outcomes that go beyond some of the traditional, bioactive lipids that we've measured previously. So one of the first studies that we've conducted using these lipidomic biomarkers was to evaluate which of these metabolites were associated with pregnancy outcomes. And I've highlighted spontaneous preterm birth again here as an example. And this plot here is a volcano plot where the x-axis is the odds ratio for spontaneous preterm birth, and the y-axis is the negative log 10 p-value. And we've highlighted the signals and labeled the signals in red that are robust to a false discovery rate of 0.15. So what we've learned in this study was that higher circulating concentrations of plasmonyl phosphatidylethanol were associated with higher odds of spontaneous preterm birth. And these associations are interesting because mechanistic studies have found that this group of phospholipids are involved in neurodegenerative disorders and cardiovascular disorders, and these potentially could be mediated through chronic inflammation. We also observed that unsaturated free fatty acids were associated with higher odds of spontaneous preterm birth, and these may be indicative of catabolism and breakdown of larger lipids. So these findings provide additional insight into these bioactive lipids, pregnancy outcomes, and hopefully in the future, when coupled with other multi-omics domains, we can learn even more precise biological mechanisms that are related to these particular biomarkers. So most recently, we just submitted a preprint for a study where we were able to evaluate these lipidome-wide metabolites as potential signatures of prenatal phthalate exposure in the PROTECT cohort. And this figure here highlights one of the main findings from the study. This is a Manhattan plot where each of the phthalates is on the x-axis, and on the y-axis is the negative log 10 p-value. And each of these point estimates are single lipid metabolites that we're quantifying. And what we learned is that among all the different signatures that were noted, one of the strongest signals that appeared multiple times with different individual phthalate metabolites were these signals with diacylglycerols. And based on what we've learned from the literature, these signals with diacylglycerols is biologically consistent with studies that have focused on phthalate exposure and that have reported that phthalate exposure can alter lipid catabolism and the regulation of triglyceride catabolism as well as lipase activity. So an important future step will be to integrate this multi-omics domains to determine if there's consistent biological plausibility in these associations with phthalates. And this also, it expands our understanding of how phthalates might be acting through lipid-mediated processes. So each of these single and multi-pollutant mixtures analyses that we've applied so far demonstrated several different important associations with intermediate biomarkers. And therefore, one of the additional next steps that we've had the opportunity to explore is extending that evidence to formally test for mediation analysis, particularly with these bioactive lipids and the prenatal exposome. So mediation analysis is an important causal inference tool that allows you to disentangle the proportion of the total effect of an exposure on an outcome that's attributable to some mediating variable, in this case, the bioactive lipids that we've been exploring. But one of the major challenges in mediation analysis is that there is still much needed innovation, particularly in approaches for analyzing multiple exposures and multiple mediators simultaneously. So in my postdoctoral fellowship, that I pursued different approaches to ameliorate this challenge. And we developed this methodological framework that integrated multiple different methods to evaluate a mediation analysis setting that incorporates chemical mixtures as well as multiple mediators simultaneously. And I won't go into too much detail on each of these methods, but I will just highlight that an important contribution that we made in this framework was proposing a hypothesis-driven dimension reduction approach where you reduce the dimensionality of the exposure matrix and mediator matrix based on literature-derived grouping structures. And so this can be, for the exposure matrix, this could be chemical classes. For the mediator matrix, this could be biological groups such as these enzymatic pathways, or if you're dealing with different omic scale data, you can do other different clustering approaches as well. And what we learned from this approach is that these whole class level analyses, chemical class level analyses with whole mediator groups really allowed us to evaluate whole pathway effects rather than single biomarkers. And this is particularly important because oftentimes some of these biomarker signals can be particularly sparse, especially in exploratory and smaller data sets. So going forward, I will be advancing this body of work in mediation analysis to enhance our framework to investigate these bioactive lipid mediation signals between the prenatal exposome, but now focusing on infant neurodevelopment. And I'm excited to share that this work has been recently awarded by the Environmental Influences on Child Health Outcomes program. And I will be integrating data across 3 different echo cohorts, the PROTECT cohort, as well as the chemicals in our bodies cohort, which is based in San Francisco, and the Illinois Kids Development Study. So to summarize this work, I have been, the past work that I've been showing you illustrates thus far that there's compelling evidence that polyunsaturated fatty acids and their eicosanoid metabolites are predictors of spontaneous preterm birth. And this is still evident across more than one prospective birth cohort. And going forward, we will continue to analyze these biomarkers in larger samples to further leverage their utility in predicting additional maternal and child health outcomes as well. The second takeaway here is that we've had substantial evidence now that toxicant classes exhibit differential signals with endogenous biomarkers, particularly bioactive lipids. And this is likely a function of unique molecular mechanisms of action. And this really necessitates application of a comprehensive ensemble of mixtures analysis approaches in order to gain greater confidence in some of these associations across different biomarker groups. And finally, the third takeaway from my presentation is that now that we're starting to see this expanding measurement of omic scale data, when we applied this in the lipidomic setting, we see that this yielded greater insight into potential mechanisms, and this can provide you with even greater details, particularly with phthalates so far, of different lipid-mediated pathways that might be affected by human exposure. So with that, I would like to acknowledge the study participants in the Life Codes and Protect cohorts that I've had the privilege of conducting research in. I also want to acknowledge my incredible network of collaborators and co-investigators. And finally, the different institutional and government, as well as foundation grants that have been critical for funding this really exciting research. And with that, I will conclude my presentation and open the floor for any questions.
Thank you, Max. That was really great. Does anyone have questions? I have questions, but if anyone wants to jump in. Okay, I'll loosen people up here, Max. So this was super interesting stuff I hadn't really heard about before. What do we know in terms of how much this might help us explain huge disparities in preterm birth that we see in the United States with Hispanic and non-Hispanic white women having much lower rates of spontaneous preterm birth compared to, for example, non-Hispanic Black women in this country? Do we know if there are differential levels of these fatty acid biomarkers by race, ethnicity, or socioeconomic status?
Yeah, so far, well, the life codes cohort is predominantly white and higher educated study sample, whereas Protect has different demographic features. They're marginally marginalized community, and we do see some evidence of different levels of these bioactive lipids, and this is to be expected given that there are different, especially like different dietary factors could be influencing these lipid levels. And so yes, that going forward, that's a really important concept that we're trying to evaluate in our statistical modeling, especially like in that prediction analysis where we are trying to predict spontaneous preterm birth in Protect, but we were training the data on the life codes cohort, right? And so I think there are different ways. And I'm still learning about the different ways to develop sort of transfer learning frameworks. And one of the ways that we can do this is by, especially as we gain larger sample size, is one of the ways we can do it is by enriching the training data set with a mixture of the underserved community participants and sort of helping the training model, have greater accuracy by doing that sort of mixture of the study participants.
Cool, that's interesting to hear. Others, oh wait, let's see. Nope, just congratulations from people, not questions in the chat. I thought maybe there were questions in the chat. I have more questions though.
Should I stop sharing?
So yeah, I'll ask one more question. How do kind of the effect sizes that you're seeing here compare to something that folks might be more familiar with, like a 10 microgram increase in PM 2.5 exposure or something? Like, how important does this exposure, this biological pathway seem to be in relation to something else? And also, what does the shape of the exposure response curve look like? Have you guys looked at that?
So I'll just focus, for example, like on the mediation model, like with phthalates, for example, the level of effect size with the outcome. I think we were seeing like phthalates were associated with a 1 week lower gestational age at delivery, which is a pretty sizable proportion of, or pretty sizable effect. We saw that in that effect, there was about a 15% mediation through the cytochrome P450 pathway. So, it is really difficult to put those numbers into like effect size comparisons, but it is that sort of relative interpretation, right? So like we're seeing that, 15% of that effect could be mediated by this pathway. And so I think the major importance of knowing that is that now that you've learned this pathway is being targeted, you could potentially detect that signal earlier on in pregnancy and have better informed understanding of the risk that that person might experience throughout their pregnancy. So that's like sort of like the interpretability aspect of these particular findings.
Okay, that's helpful. Because I guess the other thing I was trying to think about was kind of what the distribution of this looks like across people, and whether there's like upregulation of this pathway, if there's a threshold at which it becomes important, or if it's always important. Just kind of getting a little bit better sense of that kind of information.
Yeah, I think that so that's like very much, that is a really important next step in terms of because okay, well, these biomarkers are very much emerging, like these icosanoids. There are not that many laboratories that are measuring this
Okay.
extensive of a panel. And so like when I conducted that that study with the icosanoids and the preterm birth, I I really I don't remember there being any other studies at the time. There were studies that looked at the parent fatty acid compounds like arachidonic acid, but those metabolites, I don't think any had focus on them. So, to some degree, we're at the cusp of like trying to figure out where those thresholds are. And I think building these prediction algorithms can start to chip away at that a little bit by seeing like, okay, at a certain threshold, is there sort of an even higher prediction? And that's something that, I think collaborating with clinicians might be a little bit helpful as well. But it's really hard with these smaller study samples or with these pilot exploratory sample sizes.
Yeah, you want this like from NHANES to understand kind of
Yeah.
what this really looks like. But no, exciting to be the first person to publish on it. So very cool to have you present such novel work. Awesome. Do others have any questions? People are real zoomed out come end of July, year and a half into the pandemic.
Hi, thank you for your talk today. Which neurodevelopmental outcomes are you considering to look at?
Yeah, so in this new project, I'm going to be focusing on this early measure that's quantified through its non-nutritive suck measuring device that evaluates the infant's sucking patterns, basically like in the first few weeks of neonatal life. And so that's one of the parameters I'm going to be focusing on. And it's an indicator of motor function. And the other parameter that we'll be focusing on is infrared eye tracking, which I think is quantified a little bit later in the neonatal period, maybe 6 to 7 months.
Okay, thank you.
Yeah.
So Max, what's like 4 more steps down the road after you do this? What's the what's like the big picture thing that you're excited about?
Yeah, so I feel like gosh, it's so funny because I feel like I've been working with these biomarkers for like the past 2, 3 years, but I feel like I've barely chipped the surface of them, and particularly some of these like methods that we've been working on. So I think going forward, big picture, I'm really excited to expand the utility of these biomarkers. So in particular, I just worked on this R1 proposal as MPI, and we're applying these bioactive lipids to explore their predictive capacity for maternal depression during pregnancy and postnatally. And so, there is some emerging evidence that these lipid biomarkers are indicative of, neurological signaling, And this could potentially result in sort of neurobehavioral outcomes as well. So it's really exciting. And that's one emerging area that I'm really excited to focus on.